Martinell from Minoryx Therapeutics. He's going to give us a clinical update on the different programs that they have. Welcome.
Okay, thank you very much. It's a pleasure to be here today. It's my first occasion in this conference. My colleagues attended in the past, and it's great to be here. First of all, congratulations for all what you are doing and doing the newborn screening. Great progress in the US. Hopefully, Europe will follow one day. Also, a special thanks to the patients that have been participating in our studies. Thanks to understand that being in a clinical trial is a burden. It requires an effort, commitment, and without that, our work would not be possible. So many thanks in there and your persistence. And also to the investigators, sub-investigators, clinical trial coordinators from the various clinical sites. Also thank you for everyone, because without that support, I would not be here talking about leriglitazone. And before moving to leriglitazone, just as a disclaimer, so this is not an approved drug, it's an experimental drug still. So we are still in clinical trials and we have not managed yet to get it as an approved drug. So all what I'm going to say, you need to take it from that perspective of results that will need further investigations and of course the regulatory evaluation of them. For those that don't know Minoryx, Minoryx is a company from Barcelona, founded now many years ago. Focus in the ALD, that's where we are focused with leriglitazone, trying to make it with this drug for this disease, although we also have an interest in other similar indications to ALD, orphan disease, rare disease affecting the brain, and where the medical need is very high. And leriglitazone is an oral treatment. In this case, is a molecule that we call as a PPAR gamma agonist. I'm not going to enter into the details of that, but this is a molecule that reaches the brain, and in the brain has an effect on inflammation, myelination, mitochondrial function, and these are all aspects that are affected in ALD. Also in other indications, so that's why I mentioned that there's the potential for other diseases other than ALD, but particularly in ALD, these are important elements that are related with disease progression, and that's why we believe that the drug could help in this indication. Maybe you are aware of the regulatory status of the drug if you are following the field and the news. As you may know, in Europe, we requested a marketing authorization application already, and we got initially a negative opinion out of that. It was an application that was trying to cover broadly the entire ALD population, but we are now trying through a re-examination process, that the name of the process essentially is another opinion that we are going to get during this year with a special focus on cerebral ALD. And that's why today I'm going to focus more on this part of the data that we have. Not only because in Europe, this is where what we are discussing with the EMA, but also because in the US, we have a study ongoing that there's another presentation from Dr. Fatemi specifically on that study, a study called CALIX, and that the study is in adult patients with cerebral ALD. So that's also another reason why I'm going to focus more specifically in this aspect of the drug. Okay? And before moving into the data, let me show you this figure. That's actually, it's a new figure that we came up with. And the reason why we are using this figure is that because in our interactions with regulators, we found out that it's actually not always clear how lesions evolve in ALD. It's clear for pediatric patients, the classical, let's say classical between brackets, the pediatric patient with gadolinium enhancing lesions, so that is clear because there's been other development, there's the transplant, so that's pretty clear. But it's not that clear what happens before that. And it's not that clear neither what happens in adults. So that's why we try to simplify, or for the expert in the room, forgive me if that's too simple in your opinion, but we try to simplify what happens in these three boxes that you see in the screen. First, in any ALD patient, the brain may look normal. By normal, I mean no visible lesions, although we know that with more sophisticated techniques, there may be already some abnormalities, and there's been proposals of using perfusion or DTI, all these more sophisticated MRI to detect earlier than a lesion is visible. But let's
simplify it that some patients do not have lesions in their brain. Then at one point, a lesion may be visible, and such lesion may be there, not moving, or may move rapidly into a rapidly growing lesion. That is when we see typically gadolinium enhancement, not always, but typically, and that's when we know pretty well what's going to happen from that point. That is where the natural history is more clear. When patients move into the last box is when we expect clinical deterioration very rapidly and death in a few years' time, if untreated. And then in the bottom of the slide, you see what we have in our studies, because we have different studies, and that reflects how the patient population is distributed in our studies. That's what I'm going to explain. We have a study called ADVANCE. That was a study that now is completed, and the study that was in adult patients that could have no lesions, or adult patients that could have lesions in the brain, which is actually 1/3 of the population. But we specifically excluded adults that were in the last box, adults that were already with progressive CLD. Then we also have Nexus. I'm going to talk about it as well. It's a pediatric study where we have patients with CLD, pediatric patients with CLD, with non-enhancing lesions or with enhancing lesions, but all of them CLD. And now we are starting with Calyx, which is a study in adults, all of them with gadolinium-enhancing lesions. So the last box of this simplification. Let's go. So ADVANCE is the first study that I'm going to talk about it today. ADVANCE, as I said, is a study that is completed. It's a study where the primary objective was related with the myelopathy, so AMN, but we also look at the progression of cerebral lesions. This was a double-blind placebo. It's a very large study, more than 100 patients, so several here in the US, and that was already published data. And recently we completed the extension phase of the study where patients have been treated for 3 additional years, all of them inactive. Then we have Nexus. This is pediatric patients between 2 and 12-year-old with CLDs, 23 patients, open label, where we conducted already the first analysis of 6-month treatment, 24 weeks, and now we are continuing towards the end of the study, that is 2 years treatment. And that's a study that I'm also going to comment. We already presented some of the data so far, and I will show you some updates here. And then Dr. Fatemi, as I said, will talk about Calyx and the study that is now running here in the US and also in Argentina and Brazil. So be attentive to the next presentation if that's what you are interested in from Ali. So moving to ADVANCE. A few words about the design, but again, without trying to complicate things. So it was 100 patients. As I said, these are all patients with AMN. All of the patients had AMN because these adults with myelopathy, and the patients could have or not brain lesions. What we excluded, it was patients with gadolinium enhancing lesions, but about 30% had brain lesions in the MRI. The primary endpoint was 6-minute walk test, which is specifically related with the myelopathy. That was missed, as we published in the Lancet publication. We couldn't see a difference in the 6-minute. We observed differences in other elements, such as the body sway, that it's related with balance. As you know, this is an important element affecting the patient's day-to-day life, the problems of balance. But as I said, the primary endpoint of the study in this case was not met. Having said that, we also had several endpoints that relate to the cerebral component of the disease, and that's what I'm going to show in the upcoming slides. Going back to this 3-box scheme that I showed you, the most relevant clinical finding of the study, if you want, was that in this study, we observed that several patients on the placebo arm end up developing progressive CLD, so move to the last box in the scheme. And that occurred in both patients that had no lesions at baseline in the study and patients that had lesions at baseline. The lesions or not lesions, when you have lesions, the risk increases. So it was more or less double risk to move to the last box if you already show lesions in your brain. But patients that were without lesions were also showing this transition or this evolution into progressive CLD, which just reflects a very important message. It's very important to keep regular monitoring, and I want to insist on that. I know that is a burden, is year MRI, when there's nothing out there, is not necessarily something appealing. We completely understand, but cerebral ALD can occur at any time in adults. So it's important, the regular monitoring, because it needs to be detected early for the same reasons as in pediatric patients. The earlier you detect, the better the perspective if any treatment is out there. So it's very, very important, regular monitoring. That's the first important message. But what happened with leriglitazone? We did not observe any case of patient moving to the last box with leriglitazone in this study. And this data is based on the clinicians' evaluations, the diagnosis of progressive CLD following the standard of care, based on their own assessment of the MRI data, also the clinical data that was coming from the physicians. But how the efficacy outcomes of the study look like compared with this clinical finding. And in the study, we had different outcomes that were related with that, and all of them pointed into the same direction. So the drug showing in this study a reduction on the cerebral lesion progression. The figure that you have on the left, what we called cerebral lesion progression, that's purely MRI data. So it's central readers looking at all the MRI data, so same criteria everywhere. And they were saying if patients were showing a change in the MRI, yes or not. It could be a big change, it could be a small change. So that was just if there was a change. And as you see, there's also the gray bar is larger than the blue. The gray is the placebo, the blue is the active, showing that there's a lower number of patients showing changes in the MRI in the active arm compared with the placebo. The graph in the middle is the LES score, which is a scoring system that is used to evaluate the extent of the brain lesions in the MRI. There's also a larger increase of the LES score in the placebo group, in the gray bar, compared with the blue. And on the right, you have neurofilament light. Neurofilament light is something that we measure in plasma, is a biomarker that is indicative of axonal degeneration, which is related with the lesions again. So it's, if you want, another measure of lesion load, although in this case it's measured in plasma, pointing in the same direction. We see a higher increase in the placebo. And all that is driven by the fact that there are more patients in the placebo arm moving to the progressive CLD compared with the active. These results, of course, needs further evaluation, and that's part of the extension piece of the study to look precisely into that and to get further confirmation. But that was the first evidence that the drug may be doing an effect on the cerebral component of the disease. The next evidence comes from Nexus. Nexus, as I said, is a pediatric study. In this case, you have here the design is open label. In this case, all patients are on treatment with leriglitazone. We enrolled patients between 2 and 12-year-old, and that's because in this age range, based on the data that is out there, is where we do not expect many patients to arrest by themselves. So in this age range, most of the patients are expected to progress. We enroll patients with either non-enhancing lesions or with gadolinium-enhancing lesions, more or less half and half. Both populations are expected to progress in this time frame. And as I said before, we have about 20 patients. We look into the data 2 times. So we look 1 time already, and now the final analysis will be after 2 years. But the first time where we look into the data was after 6 months of treatment. In 6 months or 24 weeks is when we expect to see clearly lesion growth in this population. So that's especially what we look at, if there's a reduction into that, to decide if the trial can continue or not. Okay? That was the objective of this 6-month assessment, to see if there's an effect on the lesions. And then after 2 years, what we want to assess if the drug is able to completely arrest the disease. And by arrest, I mean that the patients do not progress clinically and do not progress radiologically neither. So we need to see both. And by radiological progression, I mean no lesion growth, no enhancement, so completely stable in this case, which is, of course, a challenging objective to achieve that. Then we have a number of other endpoints that I don't need to go into details. We already presented in different meetings and patient advocacy meetings as well, but the data of the first few patients at 24 weeks. So we presented the data from the first 11 patients that we had at 24 weeks. Now we have more patients, so the complete cohort of 20 patients that are evaluable in the study. We have that 19 out of the 20, so 95%, showed no significant lesion growth at 6 months of treatment. Significant lesion growth in this study is assessed by central readers that are also with the coordination with Martino Center in Boston, but especially with the experts in the field, such as Eric Malak, Patti Mussolino. So these are the people assessing if the things are changing or not. So it's not what I'm saying here, is their assessment. And the lesion growth at 6 months means that they look at the 6 months compared with the previous time point, which is at 3 months, and they say, from the last time point, do we see an increase or the lesions are stabilizing, particularly when we compare with natural history. There may be some little growth, but is this really different from the natural history? We see that lesions are significantly growing as expected. And as you say here, the majority of patients do show that at 6 months, there's an important effect on lesion progression. Importantly, all patients are clinically stable at 6 months, which is that, if you want, somehow expected, because we expect to see the clinical deterioration in a bit longer time frame, more in the range of a year, and you will see the data of a year in the next slide. And we also had some patients that were fully arrested, so no enhancement, no growth, no clinical progression at that time point. And now, based on this data, which is very positive from the perspective of the minimum bar that we require of patients to meet this same kind of criteria to decide that the trial continues. So now we are going for the end of the study, which is 2 years follow-up, and hopefully we'll be able to update you soon on those results. But how the data looks like in its entirety. So now you're seeing the screen all the data that we have as of today. In this slide is NFS, the score that the higher the number, the worst. So is the worst the clinical or the neurological function in this case. In the left, you have what is available from natural history in untreated patients. So you see that this score is expected to go up. In the right is the transplant data that's been published in the same score. In the middle, you have leriglitazone. So far, we are not seeing patients increasing in this NFS, and they are difficult to see the lines because all are in the bottom, but there are already 10 patients at a 1 year follow-up. It's also true that we can not say that all what you're seeing in the screen is perfectly comparable, because the data is what we have, and fortunately or unfortunately, depending on the perspective, from natural perspective angle, the data is what's available, what's been published by Jerry here in the room in 2019, but we are not going to have more data of untreated patients because, thank God, patients are being treated. And now, so they move to transplant or gene therapy, but that means that we can also compare with the data that's been published for patients that go into transplant-based approaches. That's the right-hand side in this figure, okay? That's neurological function. So far, so good, let's say. What happens with radiological perspective? That's the less score. Here you see in the right, it's leriglitazone. In the middle is Elicel, what was published for the initial cohort of patients, but it's similar when this has been extended, and the transplant. And what we can say is that we are seeing similar profiles. So we do see some increase of the lesions in the initial phase of the treatment. The tendencies of the lesions, not for 100% of the patients, but for a majority, is to move into a plateau shape, stabilizing lesions. When we look at volumes, I'm not going to show more sophisticated data, volumetrics and so, but we see the same perspective, plateau-like shape. We see an initial increase, but then after a few months, it tends to stabilize. Again, not 100% of the patients, but a majority. So similar perspective here. That's the nexus that we have so far. So we keep going on. We'll see how the study ends up. That is expected to finish in March, in a year time from now, March next year. So we are going to have the last patient reaching end of the study. And then we have compassionate use. That compassionate use is something that is being done by clinical sites. It's the physicians that are managing this. So I cannot show you details on the data here, but give you a glimpse of what we have there, because that's being in Europe, being conducted in different sites that have the experience with the drug, both in adults and pediatrics. And we have data for about 30 patients that have a post-baseline measurement, meaning that we have MRI data post starting with liraglutide to compare with. And again, without entering into details, because this is a very diverse group. The trials, you can imagine that we are targeting a very homogeneous population to be able to extract conclusions. So trials are very narrow sometimes. Compassionate use is much more diverse. We have patients with lowest score that is low, others that is very large, without clinical symptoms or very advanced clinical symptoms. So it's very, very diverse group. So again, it's more difficult to extract conclusions that I can present. But still, we are seeing that in a majority of patients, or in a number of patients, let's say more than majority, we do see same as we are seeing in the control studies in terms of how lesions are impacted with liraglutide, seeing this stabilization. Best results are seeing the earlier we treat, the better, which is not a surprise. It's usually the case, and it's also the same for other approaches, same as for transplant. The earlier we treat, the better. That happens the same, and this is particularly true for the adults with less score below 12, that's what we consider low lesion load in adults, and without advanced clinical symptoms. This is where we do see the best outcomes. And this is precisely the population that we are treating in Calyx, or that we are planning to enroll in this trial that is ongoing and that Ali will explain in more detail. So that's what I wanted to explain. Happy to take any questions if we can. So, thank you again.
Thank you for the presentation. I think this is really exciting work. I was curious what your thoughts would be about combining this with cellular therapy. So I think it's easy to consider using the drug prior to an allogeneic stem cell transplant or gene therapy or other cellular therapies. But what are your thoughts about trying to use that afterwards, sort of after that neutrophilic recovery period, when we know that patients are still at risk for lesion progression or development of symptoms?
Good question that we got a number of times based on the data that we are seeing, and that's something that at one point we will need to explore. Clearly, as you say, it's easier to see it prior, because that's the kind of drug that you can use as soon as you see a patient, and then, if approved, of course, and then treat this patient until you find the donor, or you decide to do gene therapy, or whatever decision that the physicians and the families take. But that's kind of can be a bridge for some patients. For others, where these other alternatives or transplant-based are not an option, that will be the only, if approved again. Post-transplant, the data suppose that could be something worth to explore, but that's not, I cannot say that we have data to support it, because we have not explored it. Until now, as you also need to realize that after transplant, it's a complicated setting and we don't want to mess up that part. So we need to really to think a bit more thoroughly on that, and when will be the right moment to start on that. So I cannot give a better answer than that. So that.
Thank you.
We have a question. Thank you. I'm Laurie Dang from CHOP. That was really exciting data. I have two questions, both unrelated. The first question is, can you tell me more about the compassionate use participants, and what was the highest less score where you saw stabilization of disease? And then my second question is, have you thought about expanding this product into other inflammatory leukodystrophies like MLD or Krabbe to look at disease stabilization in those populations?
To the first, the majority of patients that we have, the highest loss score is up to 15. We had some patients that were higher than that initially, but the experience that we got is that up to one point, even if you can see an impact on radiological measures, if we talk about disease, clinical progression, it's hard to see if we are doing an effect or not. Because when the lesions are large to start, maybe stopping is not sufficient because that lesion has a consequence anyway, and that clinically translates into deterioration. And we don't really know, even if we see radiological changes, if the drug is doing something or not clinically, because patients progress and it's a very complicated setting. So we will really need to compare with untreated patients. And for that population, the natural history data is not that complete that would allow us to extract clear conclusions. So it's very difficult setting in that case too. It's easier when we go into lower less score, because then we have more data to compare with, and to start we are going to have Calyx, where it's placebo control in that case. In pediatrics we have plenty of data, so it's a different setting. It's easier to extract conclusions. Second question, yes, indeed, we are into that, including some of the indications that you mentioned, doing experimental preclinical testing to see if that could work. But certainly, based on the data, it's a good rationale also to go in other indications where there are similar components, including this strong inflammatory component. And so
Let's talk.
Sure, happy to talk.
Thank you. I just had one quick question. On the slide where you showed 40% halting of the 20, is that including the boys that had enhancement and those without enhancement at the start?
Yes.
And then do you separate that out? Because one would expect boys that don't have enhancement, I would clinically expect a higher percentage of those boys would not go on, especially if you're looking at boys in that 10 to 12 range.
Yes, exactly. So the arrested disease, the number, I did not make much emphasis into that because at 6 months is less relevant. It's both populations together, so I'm not showing the breakdown. In order to be considered arrested, patient need to show no clinical progression, no lesion growth, and no enhancement. So meaning that if it was coming from population 2, it had to get negative to be considered arrested. But again, the arrested disease is important after 2 years' time, which is the primary endpoint of the study. And at that point, we expect that patients that start with non-enhancing lesions, a majority will be enhancing, so it will be progressing. And patients that start with gadolinium-enhancing lesions, of course, will keep like that with lesions growing. And both populations we expect significant lesion growth. That was been published by Eric, that in this same kind of cohort that we have in Nexus, patient lesions grow clearly in a majority of patients, and that's also one of the important aspects that we are looking at, how lesion growth compares with this very similar cohort that's being published.
I think maybe I'm alone, but to me it seems like an important distinction of those two groups, especially if 50% would really have been classified as halted at the start of the study and 40% at the mark. So if those 40%, if 30, if more than half of them had contrast on that first read, that's very different than if that 40% is all from the original 50% that really started with the halted disease. And I hope it works, I do, but I just think that that's a pretty important distinction, because clinically we would never transplant someone that didn't have enhancement, because it's much harder to know if those people are actually going to progress. I would argue, but I'm not.
Good point. Of course, when we publish everything, it will come with all the subpopulations, everything. We will be there, yeah.
I think one thing you're trying to do there, Marc, is start drug potentially before the enhancement occurs. Just to comment on that, that if you're not waiting for the enhancement to occur, so it really becomes a natural history study of the lesion. So knowing the percent of lesions that evolve to enhancement or not. And so I think that's the distinction that maybe Catherine's trying to make.
Yeah,
not simply ignoring that data, because most of those lesions that begin, we're talking about will develop enhancement, and so preventing the enhancement is still, compared to natural history, is still a good marker.
I agree. So actually, we just insist that's why the arrested part is not that important at 6 months mark. So because
in 6 months, no matter if the lesion is enhancing or not, the natural history shows that these lesions are going to grow significantly in 6 months. For those non-enhancing, what we expect based on the published data is that in 18 months, in a year and a half, the majority will be enhancing. So that's why the study primary endpoint is at 2 years, because that covers clearly both populations. If you start with non-enhancing lesions, at 2 years, we expect these patients being enhancing. So if they move to enhancing, they are not arrested by definition in the study. And if you start with enhancing already, of course, it's obvious that for everyone that these patients are progressing, they should not be arrested by themselves, except that we know that there's some patients that may self-arrest, not more than 10% in this age range, and that would be comparable. So we need to really be different from that 10% to be successful.
Yeah, and just that's one of the distinctions here is that the safety profile is such that you can decide to treat before the enhancement begins. It's a different risk benefit than transplant.
Yeah, and at the end, if approval, as always, we are not saying that that you should use one thing or the other. It's another tool that to be in the toolbox of the physician to decide how to use it and in which time, and then come back with other approaches that will be available.
Can you comment on compassionate use in the U.S. and when that might be available? How to go about it?
Available within
Compassionate use in the U.S.
In the U.S.
Yeah.
For now, we don't have compassionate use in the U.S. and we don't expect to have it in the short term by now, because the short answer is that it's quite difficult. It works in Europe and the U.S. and it makes here we cannot do it as it's too complex for the patients hours that require that. So.
I was wondering if you could comment on the patients. It's on your slide with patient eligibility criteria and secondary endpoints. You included patients with enhancing lesions less than 9. One of the secondary endpoints was eligibility for transplant. So many of those patients, I would think, would likely be eligible for transplant at study entry. Just wondering if you could comment on that population, that subpopulation.
Yes, indeed, we had several patients that at enrollment were eligible for transplant. Essentially, all that were GAT positive were eligible by criteria. These were patients that were the transplant donor was not there at that point. And actually, in the study, and I did not mention that, but that's an important point. We do not hold at any moment the transplant. So the patients can go to transplant at any point. They don't need to stay a minimum time in the study. If they want to go to transplant the day after starting the study, they can. And that happened actually in some patients. So it's just a family and physician decision, and we don't intervene there. So what we've seen is that patients that were standing potentially visible, some cases where physicians think, this patient could be candidate, but the lesion is still very early. Let's see if there's a benefit here. If not, remove it to transplant rapidly. Others were patients that were coming without a donor and were no option. We had patients going to transplant in the study. That's also another important point. So a number of patients went to transplant. Some of them, with lesions, were not growing, but they kept enhancing. So the enhancement was still there, lesions were not growing in volume, but the enhancement was there, a donor became available, decision to transplant. Fine, perfect. Others were showing lesion growth and enhancement. So as I said before, it's not that everyone showed stabilization of lesions, but so we had both situations in the study, especially when to transplant as soon as the donor became available.